Good day, everyone. I am Mark Angelo Argalendes, and I will be discussing our last topic for Histopathologic and Cytologic Techniques Laboratory, and the last topic for finals, which will be all about the Papanicolaou staining method, a cytologic staining technique. So, the learning objectives include explain the principle of Papanicolaou staining, identify reagents and explain its functions, describe modifications of Papanicolaou staining, and perform supervised practice of method. So first, let's define what is cytology. Cytology is a highly specific and accurate technique used to differentiate tumors from other inflammatory or degenerative diseases. So it is suitable for screening purposes only. Advantage includes ease of obtaining material and ease of processing. So cytology, in contrast to histology or histopathology, cytology is specifically for the study of cells, whereas histology or histopathology talks about the physiology or the pathophysiology of tissues in general. So it is advantageous due to the ease of obtaining material and ease of processing. Ano ba yung specimen sa cytology? Often, pwede yung mga aspirates. Pwede na yung mga fine needle aspirate, pwede din yung mga fluids of the body such as pericardium, CSF, or your arthrocentesis procedure. Yun sila lahat, they could be subject for cytologic studies. However, in the pap staining technique, you will find out na pwede din pala yung mga vaginal specimens. So you have pap smear, also referred to as pap test. It was developed by Georgios Nicolaou Papanicolaou, or George Papanicolaou, from the years 1883 to 1962, and he is referred to as the father of modern cytology. So, pap smear is primarily a test in gynecological cytology. Yung mga vaginal, yung mga cervical tissue specimens, they are being stained with the pap stain produced by Georgios Nicolaou Papanicolaou. So it is used for detection and prevention of cervical cancer and other cytologic diseases of the female reproductive system. So primarily again for the diagnosis or rather detection of cervical cancer. So it involves multi-chromatic staining of cytologic smear. Multi meaning many, chromatic color. As you can see here, you have pink, blue, actually dalawa lang yung nakikita nyo. But later you will find out na tatlo pala yung stain reagents used in the pap test. So it demonstrates vaginal and cervical cells in various stages of maturation. The same technique may be used to check malignancy and other specimen types, and it is a very reliable technique. So what's interesting with pap stain kasi is that it uses 3 stains, and those 3 stains has affinity depende kung ano yung stage of maturation ng cervical and vaginal cells natin. But that portion will be further elaborated in your lecture discussion. For now, let's focus sa Papanicolaou stain. Again, it is used to differentiate cells in smear preparations of various bodily secretions, of which typically it is implemented in gynecological specimens, but you can also use it in other cytologic studies, including sputum, brushings and washings, urine, cerebrospinal fluid, seminal fluid, serous fluid, synovials, fine needle aspirate, tumor touch samples, and many cytologic specimens as well. So as I've mentioned, the Papanicolaou stain is a multichromatic stain. It involves the use of 3 stains, namely hematoxylin, OG6 or orange G counterstain, and the eosin azure counterstain, which is comprised of the eosin Y, light green SF yellowish, and the Bismarck brown Y. So, atin silang iisa-isahin. So I'm sure you're already familiar with hematoxylin. It is a nuclear stain. Bakit siya nuclear stain? Remember that hematoxylin has an affinity for acidic structures. And remember, ano ba yung meron sa nucleus? Nucleic acid. So, hematoxylin, which is basic, will then bind with the nucleus. Kaya nuclear stain siya. So, the unmordanted hematine is responsible for the yellow color imparted to glycogen. Yun yung unmordanted. Remember that hematoxylin can be mordanted with either iron, phosphotungstic acid, and even aluminum salts. So for the counter stain, which is OG6 or orange G, it stains keratin. Okay. So the original role was to stain small cells of keratinizing squamous cell carcinoma, such as in the case of sputum. So yung orange G, siya yung nagbibigay ng slightly yellow but more of orange distinction because it has an affinity for keratin. So now let's look at eosine azure, of which it is comprised of three dyes, yung eosine Y, yung light green SF, and the Bismarck brown. So the number denotes proportion of dye, such as, let's say for example, eosine azure 36, 50, and 65. So yun siya yung mga ratio. Contains two mutually incompatible chemicals, the Bismarck brown and the phosphotungstic acid. Now, they precipitate each other. So meaning to say, while Bismarck brown is a stain, Bismarck brown is a stain that is part of eosine azure. Please remember this because this is often implicated in the board exam. What component of the Papanicolaou staining technique does not stain the tissue? And the answer for that is Bismarck brown, because Bismarck brown together with phosphotungstic acid is precipitated. Kaya siya yung non-staining component of eosine azure. So what are the composition? So for eosine Y, it stains superficial, epithelial, squamous cells, nucleoli, cilia, and red blood cells. So again, ang eosine Y is superficial epithelial cells, whereas ang light green SF yellowish, they stain the cytoplasms of other cells, including non-keratinized squamous cell. Ano nga yung stain natin for keratin? OG6. However, ang light green SF yellowish natin, they are expensive and difficult to obtain. So, fast green FCF is used as an alternative, however, it produces visually different results and is not considered satisfactory by some. So, yun lang talaga yung downside. Medyo expensive ang light green SF yellowish. And lastly is yung Bismarck brown. And as I've mentioned, Bismarck brown is precipitated by phosphotungstic acid. So, if you're going to be asked, ano nga yung component ng pap stain that does not stain the tissue, that would be Bismarck brown. Now, meron din tayong tinatawag na modified Papanicolaou's technique. Sa modified Papanicolaou's, they just remove Bismarck brown. Question, ano ba yung ginagamit natin for routine pap stain? Is it the traditional or the modified pap? The correct answer is, we still use the formulation containing Bismarck brown. So, yung traditional pap stain pa rin yung ginagamit. Okay? So, for the Papanicolaou stain, stained specimens display hues from entire spectrum. So, they appear red, orange, yellow, green, blue, and violet. So, chromatic patterns are well visible, and cells from borderline lesions are easier to interpret, and producing better photomicrographs due to the multicolor attributed to different stains, and the cells are rendered very transparent. Even thicker smears with overlapping cells are visible. So to better appreciate a result of a Papanicolaou's technique, let's look at images. So ito naman ang affinity ng mga cellular structures to the certain stains resulting in the characteristic color. So yung cell nucleing natin, remember that it's rich in nucleic acid. Ano nga yung nuclear stain? Hematoxylin. Thus, it appears blue to black. Keratin and glycogen are stained by OG6, that's why they appear yellow to orange. However, superficial cells would actually appear more orange to pink as opposed to keratin and glycogen. Intermediate to parabasal cells would appear turquoise to blue, and metaplastic cells would often appear both green and pink. And remember, this is because each stain has a different affinity for each of these characteristics. So this is what cervical cells typically appear. Ito yung mga layers nila. You have your small parabasal and basal cells, large parabasal cells, small intermediate cells, large intermediate cells, and the superficial cells. So, ito yung appearance nila. Ito yung parabasal cells. Most likely, it has been stained by the eosine component. Etong pink din, yeah, it's still the eosine component. And then for the intermediate cells natin, most likely they are stained by the hematoxylin. And remember that all of those is because of the presence of multiple stains. Okay, kaya nga multichromatic siya. Remember that eosin Y contains what? LF, green. Meron din siyang eosin Y comprising the yellowish stain, and meron din siyang Bismarck brown. And then meron din hematoxylin which stains acidic components and others. So itong nakikita ninyo dito, class, yung mga small blue na mga organisms, those are actually vaginal flora. So again, this is the appearance of different normal cervical cells. Ito yung superficial cell, ito yung intermediate cell, parabasal, basal cells, endocervical cell from the side, and endometrial cells. Okay? So this is just to better appreciate the cervical cells. Another is a vaginal smear showing coccobacilli of bacterial vaginosis. So in this case, you can actually use the Papanicolaou stain to diagnose bacterial vaginosis, wherein there is an overgrowth of Gardnerella vaginalis. Ano ba yung tawag sa vaginal cells that is coated with coccobacilli? Yun yung tinatawag natin na clue cells. Bakit clue cells ang tawag? Because they are a clue that there is a pathology. Na bacterial vaginosis. So again, ito yun sila. So this is another image. The problem with Papanicolaou stain is that it is not standardized. Formulation varies among different manufacturers, and several modifications subtly differ in dyes, ratios, and timing process. So paiba-iba siya. Yun lang yung problema. Paiba-iba. So you also have the cytocolor method by the Merck Millipore International. So they have the Zepanix modification of the classical pap stain. So itong modification na ito, it's better because it provides early recognition of female genital carcinoma, gives result on malignancy, hormone status, and vaginal flora, and other specimens may be used. Actually, this is not quite different from the traditional pap staining, but the Zepanix modification produced by the Merck Millipore International provides consistency, provides standardization. And this is the procedure using the cytologic standard stain, the Zepanix modification for cytocolor Papanicolaou technique. And that covers our last topic for histopathologic and cytologic techniques laboratory. Thank you for listening.